Part 2 of your gene expression chapter 10 notes. So gene control in eukaryotes is kind of unique. They don't really go into too much detail specifically, but they give you some examples of gene control in eukaryotes. One of them has to do with the X chromosome, and I didn't know this before I did this lecture. The X chromosome has the ability to be inactive during development. So X chromosome inactivation balances expression of X chromosome genes between female, which are XX, and male, which are XY, only in mammals. Basically what happens is one of the X chromosomes condenses, you know how you have two, one from mom, one from dad. Well, one of them randomly is chosen to be condensed and forms what they call bar bodies. And this happens when the embryo is about 200 cells big, and the cells will independently choose which X, either the maternal or paternal chromosome, will condense or not be expressed. And that's kind of really cool and interesting because if you think about it, you're 200 cells big, and then every cell, let's say it's 50-50, 100 of those cells will express the maternal X chromosome, and 100 of them will express the paternal chromosome. But those are chosen at random. So you end up with like patches and groups of cells in your body that will express one chromosome and patches and groups that express the other chromosome. And I was like, what? So an example of that is the calico cat, because you can see how it has some orange and some black, so parts of it are expressing the female maternal X chromosome and others are expressing the paternal chromosome X that you got from your dad, or that the cat got from its dad, not us. Another one is on the Y chromosome. The SRY gene expression triggers the development of male traits only in mammals. So when this gets turned on, then the male traits come out, and that's found on your Y chromosome. And in plants, there's this expression called the ABC floral identity genes, and governs the development of specialized parts of a flower. So basically what happens is A turns on, and then B turns on, but A is still going on, and then so there's some overlap. So A causes sepals, and then A plus B causes something, and then B by itself causes petals, and then C turns on and A turns off. So then you've got like B overlapping C for a while, which causes parts of the reproductive parts to happen, and then C is on by itself. So through this segue of domino effect of genes and some overlap going on there, you end up with all the parts of the flower developing, which I thought was pretty cool. However, in prokaryotes, it's a lot different. It's not going to let me get off here now. Here we go. In prokaryotes, they don't have all the chromosomes that we have, so the genes are kind of like scrunched up pretty close together. And so they have things called promoters, and we'll just get into it as we go. So prokaryotic organisms do not undergo development. If you think of it, a bacteria doesn't have to develop like a human does. It's already fully developed pretty much as soon as it divides. So there are no master genes that control cell differentiation and development. But they do have to respond to their environmental fluctuations, and they do that by adjusting gene expression. So whatever's going on, whatever nutrients are available out in their external environment, they have to adjust those genes to handle those. So when present, the bacteria begins transcription for use, and when absent, the transcription stops. So how do they do these? Well, they have operons. And operons are groups of genes that are controlled together. And operons are found in all organisms, but they are very much found in bacteria. So I've got a picture up here, and the operon is this group of lac enzymes, and lac enzymes break down lactose, so this would be present if you had milk. And you have promoters, which is the region that the RNA polymerase will bind to. In front of that promoter region is the cap site, which promotes the binding, and after that region is a repressor, which will block the RNA polymerase from actually transcribing. So a couple of things can happen. If the bacteria senses lactose present, then it'll have that cap site that starts. RNA polymerase will bind, there will be no repressor, and it will keep going and transcribe all three of these genes, because those three genes that are there together all have to be present in order to break down lactose. You actually they go in order I don't remember which one comes first, but you have to have one and then the next and then the next, and they all control enzymes that are responsible for breaking down the lactose. But if the repressor is there, let's say there's no more lactose, so a repressor binds, and then RNA polymerase will stop transcribing that to make more proteins to break down the lactose. So bacteria have to be really efficient in their system because they don't have a lot of space or a lot of extra ATP to go around. Remember, they don't have mitochondria to make this in plenty. Let's see, where was I? So genes that are used together occur sequentially on a chromosome. I think I kind of talked about that. Single promoter precedes the group of genes, so all are transcribed in one RNA strand. Now in a eukaryote, each one of these would have its own specific RNA strand, but in prokaryote, because you need all three of them to do the job, they just transcribe them all on the same page. Basically, you don't get separate papers for it. So controlled all in one step, the repressor binds to an operator to keep that from happening if you need to control it. And so the example was the bacteria lac operon allows the expression of 3 lactose metabolizing proteins only when lactose is present. 2 operators flank the promoter are binding sites for a repressor that blocks transmission. And I talked about that. You have one on either side and it can cause it to stop. Interesting fact, most adult humans do not produce the enzyme that breaks down lactose, and the ability ends in early childhood. So you might not be totally lactose intolerant, but you might be lactose sensitive to where as you get older, you're like, ooh, I got pretty gassy and had some stabby pains after I had that ice cream. That's probably your body saying, whoa, cut out the ice cream, dude. Like, we don't need that much dairy. We can handle a little bit, but not that much anymore, because your body stops being able to make that enzyme anymore. It stops expressing it because it doesn't need it as you get older. Last but not least, inheritance of gene expression patterns. So sometimes the expression of genes gets passed down from one generation to the next. And you think, oh, that only the things that are in sperm and egg get passed down, and that's true. But the genes that are in your sperm and egg can be methylized, can be changed to be expressed or not. Especially in sperm, you have a lot of RNA that gets added and modified and put back in as it goes through its process before it is fully developed. And so they found, especially in sperm and especially in sons of fathers, the expression of genes can be passed on, which is crazy. The genes are there, obviously, but the ability to express them at a higher level or a lesser level gets passed from one generation to the next. So those are called epigenetic changes. So they're heritable modifications to DNA and its function, but not the base sequences. So it modifies the DNA because once a piece of DNA gets methylized, I'm saying that wrong today. I don't know what's wrong with my brain. It doesn't come off. It stays stuck there. It's like that sticky Lego that you can never pop off. One little one that gets stuck, you know what I'm talking about. That's what happens to this DNA. So it is more permanent than the methylation of histone proteins if you methylate DNA. And it will transfer to all of that DNA's descendants. So if your germ cell for your sperm or your egg gets methylized, I know I'm saying it wrong, I'm sorry guys, but then all of those descendants would get that result. And this is found in C to G base sequences. So if you have like a C and a G together, like this, hold on, I'm trying to write, it's not letting me, but a C over a G, nope, it's not letting me write anywhere, I'm so sorry, on one side, and so on the other side it would be G over C. Whatever happens to that C on that side will also happen to the G on the next because it will read it both ways. So wherever C pairs with G, it can be methylated. The example is grandsons from a Nazi famine. They found that these grandsons were able to live 32 years longer than grandsons from people that did not survive the famine, and they took into account all social, economic, and all this other stuff. So that was pretty cool. And smoker sons, the sons of smokers, were found to have a higher rate of obesity and diabetes, I think. I know it was at least obesity. I don't have the book in front of me. But then non-smoker sons. And again, they accounted for all the other factors. So that's interesting. Be healthy when you go to have babies, guys. It's very important that you pass on good DNA. DNA methylation results in differentiation and is essential to embryonic development, silencing genes that are no longer needed. Think stem cells. So like you don't keep stem cells in your body permanently. Remember, your cells lose the ability to differentiate as you grow. And so they get silenced. It's not just expressing, but it's also silencing genes that are no longer needed. So you can't turn them back on after they get methylated. I know that's wrong. Anyway, you got the idea. So there's your notes for Chapter 10, Gene Expression.